The central dogma of molecular biology. In asexual reproduction, often referred to as mitosis, a parent cell divides to become two daughter cells. Before this occurs, the nucleus of the parent cell must copy its genetic material. It does this during the phase called synthesis in the cell cycle. This happens during interphase. It does this so each daughter cell has an identical genome as the parent cell. Furthermore, DNA is required for the synthesis of proteins and for the functioning and growth of an organism. Hey Srin,
What?
what did the DNA strand say to the other DNA strand when it was mad at it?
I don't know. What?
Stop copying me! DNA stands for deoxyribonucleic acid. There are 4 monomers found in DNA: adenine, cytosine, guanine, and thymine. DNA comprises of nucleic acid, which is comprised of nucleotides. Nucleotides are a compound consisting of a nucleoside linked to a phosphate group. A nucleotide consists of deoxyribose sugar, a phosphate molecule, and a nitrogenous base. A nitrogenous base is like a flat ring in the middle of a DNA strand. Their function is to spell out the sequences of functions called genes. As mentioned before, a nitrogenous base consists of 4 different bases within it: A, C, G, and T. In the DNA, adenine only binds with thymine, and cytosine only bonds with guanine. All the nitrogenous bases are hydrogen bonded. DNA comes in a structure called the double helix, 2 strands twisted around each other. It has an anti-parallel nature, which means that the top strand of DNA replicates from 5' to 3', while the bottom strand of DNA replicates from 3' to 5'. What does this mean? Sugar in the form of carbon molecules make up DNA. Carbons are numbered in a clockwise direction. The 5' carbon is outside the ring structure for both strands. The top strand is called the leading strand, and the bottom strand is called the lagging strand. DNA can only be replicated continuously in the direction of 5' to 3'. This will make more sense once we get into how DNA replicates. There are many important components in DNA replication. Firstly, helicase, an enzyme, is known as the unzipping enzyme. Helicase breaks the 2 strands of DNA apart, therefore breaking the hydrogen bonds. It is to be noted that 2 DNA strands are not identical, they just complement each other. Next, DNA polymerase, another enzyme, replicates DNA molecules to build a new strand of DNA. Primase, another enzyme, is known as the initializer. Primase can't do its job without something called a primer, a short sequence of nucleic acids. Primase makes the primer first. Ligase, the gluer, helps glue DNA fragments together. DNA replication begins at a point called the origin. There can be multiple origins within a DNA. Helicase unzips the DNA strands, like you would unzip a zipper. SSB, or single-stranded binded proteins, bind to DNA strands and keep them separated. Primase creates a primer on both strands. This is essential because the DNA polymerase needs primer to know where to begin the process. The leading strand, or the strand that goes from 5' to 3', is the strand that the DNA polymerase can use. This refers to the directions in which DNA is replicated. The DNA polymerase can only replicate continuously in this direction. For the lagging strand, the DNA polymerase creates fragments of replicated DNA, called Okazaki fragments. The primers, or the initializers, have to remove their RNA base and make it into DNA. The ligase, or the gluer, takes care of the gaps in the Okazaki fragments. This results in 2 double helix strands of DNA. This process can be classified as semi-conservative replication, because each copy contains 1 original strand and 1 new one. Now that the DNA is replicated, how can it be used to code for your genetic traits?
Why was the libraries working so hard?
Why?
Wanted to make ends meet.
Transcription is when genetic information from inside the nucleus is brought to ribosomes so that proteins to enact those instructions can be made. Transcription is part of a process called protein synthesis, the process where DNA makes proteins. DNA sends this message to the ribosomes, the cell organelle which creates proteins, through mRNA, a type of RNA. First, let's talk about the differences between RNA and DNA. RNA and DNA are very similar, but there are some key differences. RNA stands for ribonucleic acid, while DNA stands for deoxyribonucleic acid. They're both nucleic acids, however, DNA is missing one oxygen. This allows for DNA to be more stable, which is important as it carries genetic information in one place for long periods of time. Another difference is that DNA has thymine, while RNA has uracil instead of thymine. Uracil pairs with adenine just like thymine does. The first step of transcription is initiation. It occurs in the part of the DNA strand called the transcription unit. This is how transcription starts. The enzyme RNA preliminary begins to scan the DNA and binds to a space called the promoter site. It unwinds the two strands in the double helix. Elongation is the next step of transcription. It starts after the DNA bubble opens. This is where mRNA is made. Each base of one strand of the DNA starts to bind to its complementary base to create a strand of RNA. In RNA, adenine binds to uracil instead of thymine. The strand of RNA is identical to the non-transcribed strand. This is why the non-transcribed strand is called the coding strand, while the transcribed one is called the non-coding strand. The last step is called termination. The process stops when RNA preliminary reaches the termination point. The open DNA bubble closes up and the mRNA detaches from the DNA. The mRNA is a copy or transcript of the genetic information in the gene. The mRNA can now go outside the nucleus to deliver the message to the ribosome and begin the next process of the protein synthesis, translation. In almost all eukaryotic genes, coding regions called exons are interjected by non-coding regions called introns. After transcription, during a process called splicing, the introns are cut out to create a mature strand of mRNA. Hey, why was the RNA so hormonal before translation?
Why?
Because it was being made into a protein. Deciphering the genetic code. Each codon is made of 3 nucleotides, and each codon codes for an amino acid. There are 64 possible sequences of codons that can be made with the 4 bases. These 64 codons code for a total of 20 amino acids. In order to identify which codon codes for which amino acid, look at the drawn chart. tRNA's main function is to carry amino acids to the ribosome to incorporate into the protein. tRNA's basic secondary has a very distinct structure, with the shape of a clover leaf. The arms are formed from the hydrogen bonding between the base pairs. The area where 3' and 5' are paired is where the amino acids attach. The part that is labeled as an extra arm is because it sometimes varies in length. The D and TYC arm are named as such because of the presence of modified bases. The molecule then adopts a tertiary structure, looking something like an L, when the molecule folds and base pairing occurs between the arms. As we have covered before, there are 4 nucleotides in RNA, A, U, C, and G. 3 nucleotides form a codon, which is another name for an amino acid. Codons are part of the mRNA. The mRNA, also known as messenger RNA, carries the genetic code from the DNA to the ribosomes. An anticodon is complementary to a codon and is part of the tRNA. Each codon has a specific anticodon, and during translation, the codons and anticodons form appropriate hydrogen bonds. After the transcription to RNA is finished, it must be translated into proteins. The first step of translation is called initiation. In this stage, an initiator tRNA, a special kind of tRNA, starts protein synthesis by binding to a small ribosomal subunit. Then joining of the first codon to anticodon occurs, and the start codon is usually AUG, which codes for methionine. The next stage is elongation. In elongation, the string of amino acids lengthens to the growing protein chain. The order of the amino acids is determined by the sequence of codons in the mRNA. In elongation, the polypeptide chain is synthesized, or produced as the mRNA passes through the two ribosomal subunits. The tRNA molecules then move the amino acids to the ribosomes. Then, in termination, the ribosome reaches the stop codon, releasing the mRNA transcript from the ribosome. The polypeptide chain is also released from the ribosome. Translation is complete. A mutation is when there is a change in genetic material in a nucleic acid, either DNA or RNA. Mutations usually occur when the wrong bases match up. How terrible would it be if adenine matched up with cytosine and thymine matched up with guanine? Other external factors that might cause mutations are chemicals and radiation. Some internal factors that might cause mutations are errors in DNA replication during interphase. You might think that all mutations hurt organisms, but that's not true. Many mutations can in fact help organisms. One type of mutation is a gene mutation. Our DNA makes up our genes, which gives us traits. When the structure of the DNA changes, it can influence the proteins being produced and the traits we receive. There are many ways the proteins can change. First is substitution. This is when the wrong bases match. Next is insertion. This is when an extra base is added in. Next is deletion. This is when a base is removed. Insertion and deletion are the most dangerous in regards to the severity of the mutation. Bases in a nitrogenous base are read in threes, so when a base is added or taken out, all the coding that follows the bases can get messed up. This is called a frame shift. There is also a kind of mutation called a chromosome mutation. A chromosome is made up of DNA and protein. There are different kinds of chromosome mutations, including duplication, when an extra chromosome is generated, deletion, when genetic material from chromosome breaks off, inversion, when a chromosome segment gets inverted, and translocation, a fragment breaks off from one chromosome and attaches to another. In meiosis, sometimes the chromosomes don't split properly. This is called a non-disjunction. This results in one daughter cell having too many or too few chromosomes. Lastly, there are silent mutations. A silent mutation occurs when there is an error in DNA replication, but it does not cause a change in the amino acid sequence. Silent mutations also won't have any effect whatsoever on the offspring. You may have one right now and not even know it.